will be chaired by Dr. Shrikant Gattu. Dr. Shrikant Gattu is currently working as a global program medical director at Novartis, Germany, a pharmaceutical physician and digital enthusiast with over 15 years of total experience in research and drug development, combined with strategic roles in senior leadership team in global development of biosimilars at Sandoz and established medicines franchise Novartis Pharma as global medical lead for respiratory range of products. He also worked as strategy and operation leader in Sandoz Biopharmaceuticals, medical lead on oncology biosimilar development, medical lead on a LABA asthma study. He is also a business lead for artificial intelligence machine learning digital project for clinical development. And the session is also chaired by Dr. Ramesh. He is from Viatris, Bangalore. He has completed his MD Pharmacology from Sri Ramachandra Medical College and Research Institute, Chennai. He pursued his MBBS from Bangalore Medical College. He has completed CCEPDM course from Public Health Foundation of India. He is also IEC member of Sri Sathya Sai Institute of Higher Learning. He has held various positions and he has a cumulative experience of more than 15 years. He has taken up multiple roles like head of medical services at Primus Hospital, Bangalore. He was a research physician at Clinigen International Limited, Bangalore. He was also a medical and regulatory lead from Syngene International Limited, Bangalore. Over to the chairpersons. I invite Dr. Shrikant Gattu to introduce the speaker.
Yeah, thanks, Dr. Indumati. First of all, thanks to Melvin and the SRM University for giving me this opportunity. The session is going to be on interchangeability. We have already heard, in bits and pieces from the first two sessions about the interchangeability. Now, the interchangeability is a very specific regulatory term, and there are terms like substitution, etc. And we'll hear more about that from Dr. Viraj in next few minutes. So this concept in principle is very specific to US, but then there are other countries who have their own positions, what they mean by these terms, interchangeability, substitution, etc., etc. So let's hear more about this from Dr. Viraj Suvarna. I'll hand it over to Dr. Ramesh for introduction of the speaker here now.
Thanks, Shrikant. Dr. Viraj is associated with Biocon Biologics in the capacity of Vice President and Head of Medical Affairs. And he has experience of more than 23 years in the pharmaceutical industry. He has held various leadership positions at Lupin, Dr. Reddy's, and Boehringer Ingelheim before joining Biocon Biologics. And he has an excellent academic credentials. He has completed his graduation from PNMC Mumbai with a distinction and gold medal in anatomy and pharmacology. He has pursued MD Pharmacology from Grant Medical College Mumbai, and he has stood first in the university. He has also completed MSc Pharmaceutical Medicine from Iberia University, Dublin. The floor is yours, Dr. Raj.
Thank you, Dr. Ramesh and Dr. Shrikant, and of course, Dr. Melvin. I will now just share screen. You can see my slide, no? Hello?
Yes, sir.
Yeah.
Yes, sir.
Okay. So I'll straight away dive into the topic and the outline. Initially, I'll just talk a little bit about biosimilars and generics, and then about these two terms, prescribability and interchangeability. And then to give you one example, the case of biosimilar interchangeable insulin glargine, because incidentally, it was the first to get this interchangeability designation by the US FDA, and that is a matter of great pride for Indians because an Indian company managed to get this for the first time in the world. Then, of course, Boehringer Ingelheim, I think, was the second company to get it for Siltezo, which is their biosimilar, adalimumab. And I'm sure now there are other interchangeable biosimilars as well. Now, what are biosimilars? We know that obviously when you look at a generic, you have the small chemical innovator or originator. When that goes off patent, then naturally you can come in with generics. It's not that difficult because all that you have to do is show chemical equivalence. And for small chemicals, it's not that difficult. And then you do a bioequivalence study, and that's it. You don't really need to do a clinical or therapeutic equivalence study. And that is the reason when you look at price differentials, it could be as high as even 80%. But when you look at a biosimilar, and I'll talk about it in subsequent slides, it is not that easy to make a biosimilar. And therefore, it is difficult for a biosimilar manufacturer to price its biosimilar the way people price generics. So here the price differential is not much, maybe 30-40%, because the biosimilar manufacturer has to do quite a bit, as I will talk about. Now, I know there are some terms and people are wondering, what is this bio better? Because sometimes when you do a study comparing the biosimilar with the biologic, the original reference biologic, generally they do it using an equivalence or a non-inferiority design. And we all know that sometimes in these studies, it might happen that the biosimilar is actually superior to the original reference biologic. Now, in that case, can we call it a bio better? Theoretically, logically, yes, but the regulator considers a bio better as a new biological entity. So which means that company will have to do the whole rigmarole again. And obviously at that stage in the life cycle of the product, nobody would want to repeat whatever the company did for its original reference biologic. And that's the reason people don't really come in with these terms biobetters. I believe there are one or two examples. I don't know, I may be wrong, but trastuzumab, I believe deruxtecan and adoemtansine were actually shown to be better than Roche's Herceptin. But still, they're not really calling themselves biobetters because naturally then they'll have to really invest in the development. But this other term is important, biomimic or biocopy. And in the EMA, suppose, your proposed biosimilar is rejected by them because it is not biosimilar per their exacting standards. And they are very strict. How I would look at them is, they were the first to come out with guidelines in 2005, and there are instances where they have rejected, some companies who have proposed biosimilars. So for them, if you don't meet their requirements, they will term you as a non-similar biotherapeutic product. In the US, such products are given the term biomimics or biocopies or intended copies or non-regulated compounds. So, I mean, they are quite strict about approving a biosimilar. So therefore, it's important for any Indian company to have its biosimilars also approved abroad, just as they have their generics approved abroad. So obviously, if you have your products approved abroad, you can command a higher price, and it also helps you from a revenue perspective. So that is why I'm not entirely clear, although I heard my good friend Shubhadeep mentioning that at least with Hetero, their biosimilars are present in many countries abroad. Now, when he said many countries, I hope he included US FDA, European Union, so EMA, Japan's PMDA, Health Canada, TGA from Australia, etc. Because it's important, for Indian companies, it should not be only Biocon Biologics. Other companies also should market their biosimilars abroad, because the moment you do that, you have to really get into rigorous, not just CMC comparison, that of course, but after that, you have to generate your own data, even preclinical or non-clinical, then of course your clinical pharmacology, your PK/PD, and obviously then the phase 3. Of course, a bioequivalence is also required. So once you get your product approved abroad, I'm not saying that the Indian regulations are extremely lax or lenient. India has actually, I think, modeled its guidelines on the basis of WHO, and the entire purpose is to make that biosimilar as affordable as possible so that you improve access. So I get that. But at the same time, I'll tell you one or two ways in which I think the Indian guidelines need to be a little stricter, because otherwise you could have problems. Now, we all know this. I don't want to spend too much of time on it, but clearly there are differences between small chemicals and biologics in terms of their production, characterization, how are they administered, the manufacturing, and of course, safety concerns. And I'll talk a little more about immunogenicity in my subsequent slides. Now, this I'm just showing you. I know everyone knows what is recombinant DNA technology, but the only reason I'm showing you this is because in pharmacology, at least when I learned pharmacology, my professor told me that, the definition of a drug, it's very simple. A substance which, when introduced into, say, a human being, it only changes existing cell function. It does not impart new function to a cell. So I was just thinking, maybe now, because of recombinant DNA technology, we can actually make an E. coli, for example, or a yeast like Pichia pastoris or Saccharomyces cerevisiae or Hansenula polymorpha produce something which it has never produced before in its lifetime. So maybe now the definition of a drug needs to change. I'm just asking this as a question. It's rhetorical. And here I want to introduce this term of, when we do studies to show that, this drug is biosimilar or this drug is a generic, what it means is that now the doctor can prescribe the same, in the sense, instead of the original reference biologic or the original reference small chemical, I can now prescribe this other drug because obviously it's cheaper. And when I say cheaper, it's not only from a daily acquisition cost perspective, but naturally, if the drug is cheaper, more people can access it. And even that person who accesses it is more likely to be compliant, is more likely to be controlled, and is less likely to have complications. And that way also, there is a price or rather a cost advantage. But just because it is prescribable, does it mean that a patient who's currently on that original reference biologic or small chemical innovator or originator compound, does it mean the doctor can straight away switch that patient from that original product to the generic or the biosimilar? No, you have to do, and as Shubhadeep rightly said, you have to do a specific study. There is a specific design. We call it a 3-switch design. And based on that, if you can show that this patient who was on the original reference biologic now is switched to the biosimilar, after some time switched back to the original reference biologic, after some time switched again to the biosimilar. So it's like 3 times you do this kind of to and fro. And every time you monitor, obviously, the efficacy, safety, and immunogenicity. And if there is no significant difference, clinically meaningful difference, then obviously the regulator would say you are interchangeable, which means now, even if that patient is on that original reference biologic or small chemical innovator, now you can switch this patient to that generic or biosimilar. But unfortunately, for a generic, that is not required, because what happens is people believe that for small chemicals, once you show bioequivalence, chemical equivalence, it is taken to mean that clinically also there will be no difference. Actually, I'm not entirely sure, because I have actually come across cases where even with generics, if you switch the patient from the original reference small chemical innovator to that generic, there could be differences. But anyway, that's a matter for another debate. Right now, we'll only focus on interchangeability as far as biologics and biosimilars are concerned. And what's written in the below box, that is in terms of the international non-proprietary name, where, when you talk of biosimilars, what is interesting is that it's not just, for example, biosimilar insulin glargine. If you are interchangeable, then actually speaking, you're given a 4-letter suffix, which is YFGN in this case. So that also distinguishes this biosimilar from other biosimilars, so that, it's not extrapolatable. Just because one biosimilar has got something, it doesn't necessarily mean the same thing can be extrapolated to other biosimilars. And in any case, that biosimilar gets an exclusive period of 1 year. Now, after 1 year, perhaps other biosimilars can also somehow claim that they are interchangeable, though I believe it's better to ask them also to do that 3-switch design, and then they can also claim interchangeability. But that doesn't perhaps happen. And this is only to tell you that it's not only with biosimilars, it's also with biologics. I mean, this case, we know the J&J product Eprex, where with one batch, all that they did was, I think, change human serum albumin, that is in the stopper of those uncoated pre-filled syringes, to something called as 2080 polysorbate. It's a solubilizer. And because of that, some patients developed pure red cell aplasia. And obviously that batch had to be withdrawn and things like that. So all that I'm saying is, because see, even a biosimilar is ultimately a biologic. It's just that I'm comparing it with the original reference biologic, and therefore, if it is biosimilar, I claim it as a biosimilar. But ultimately, it is also a biologic. So whatever applies to biologics will apply to biosimilars as well, including that inherent variability that is seen with biologics, even from batch to batch, and similarly from biologic to biosimilar. So that's why it's important to rely on a company which has extensive years of experience and expertise with this kind of manufacturing. Now, this is something which I remember when I was in Pfizer and then Boehringer Ingelheim, so MNCs, I was also a part of OPPI. And at that point in time, we had to actually make a slide deck to kind of educate doctors on the differences between biologics and biosimilars. So I've just taken one slide from that deck. Now, what you'll see here is where we are actually trying to caution doctors that be careful, don't just move from a biologic to a biosimilar, because there are all kinds of problems and implications. And now over the years, since I've moved from MNCs to Indian companies, and maybe Indian MNCs, you can call them, I've actually some experience of, both sides. So all that I'm trying to say in this slide is, yes, with a biosimilar vis-a-vis a generic, there are differences. And that's why doctors are a little wary and cautious, and they need to be. And there are certain interesting aspects also regarding a biosimilar. For example, biosimilars need their own international non-proprietary name. So in this case, the 4-letter suffix, their label needs to be different. It can't be just a copy-paste where only the brand name is changed. Informed consent may need to be taken. There's a need for aggressive pharmacovigilance. So when you're reporting an adverse event, report it with the brand name and report it with the international non-proprietary name. And ideally, pharmacists should not just substitute without informing the physician, but in the US, if the US FDA has given you an interchangeability designation, then a pharmacist can actually substitute without even informing the physician, which according to me is not exactly right. But anyway, that's the law in the US. So this is just to tell you this variability, which is inherent, of course, with biologics. And I'm showing you both between batches, different batches of the biologic, and also between a biosimilar and the original reference biologic. There could be this variability, and therefore batch to batch consistency is very important. And therefore, if you put your faith or trust or belief in companies which have years of manufacturing experience, the science, scale, scope, technology, and years of patient experience, whether it's recombinant human, now I'm only talking about Biocon and specifically recombinant human insulin or biosimilar insulin glargine, where quite a few doses have been dosed, a lot of patient exposure days, and also in terms of the largest or one of the largest insulin suppliers by volume. Now, this is important. Last year on World Diabetes Day, the International Diabetes Federation, normally they come out with a theme just for 1 year. But this time they came out with the theme for 3 years. And the best part was the theme is access to diabetes care If not now, when? And why is it interesting is because last year was the 100th year centenary for insulin. And yet we have data saying that 33 million people with type 2 diabetes mellitus do not have access to insulin. And by another 8 years from now, the number will reach 41 million, most of whom are in Africa and Asia. So clearly we need to improve access. And maybe one way of improving access is providing quality biosimilars that have been world approved. That means major market regulators which are extremely stringent in the way they approve biosimilars. And of course, consistency. Now, this is something that even Shubhadeep talked about, so I won't dwell too much about it except to tell us that this fingerprint, like, how we are unique individuals because our fingerprint, our lip print, our tooth print are absolutely unique. You can identify people based on these prints. So similarly, this biosimilar can be identified by its print, the fingerprint. And to that extent, obviously the original reference biologic will also have its own fingerprint. And to that extent, that is why people, of course, this is not the only reason, but to use this term bio-identical. So the basic amino acid sequence may be the same, but as Shubhadeep was saying, in that process of manufacturing, then certain things happen, like, glycosylation, sialylation, amidation, acetylation, phosphorylation. And therefore, ultimately, the end product may not be identical, but therefore, you need to do studies to show that at least clinically, is there any meaningful difference between the original reference biologic and the proposed biosimilar? And as long as there's no difference, then I guess you can use the biosimilar instead of the original reference biologic. And if you do that 3-switch design, you can also interchange or switch. Now, this is a very interesting slide. I know Shubhadeep has shown it to some extent. I'll just quickly go through it. That on the left, you have this inverted pyramid that talks about the originator development, where the biggest chunk of that pyramid is your phase 3 right on top. But look at now, for a biosimilar, it's the erect pyramid. The biggest part is right at the bottom, which means in vitro studies or analytical, structural and functional characterization, which you saw on the earlier slide. And even Shubhadeep very rightly said that this is the crux, much more than even your clinical studies. So as long as you can show on a CMC basis, and I heard one question in India, are people really great at, looking at CMC data and then satisfying themselves, ascertaining that, yes, on a CMC basis, this biosimilar is similar to the original reference biologic. If that happens, then that itself is a big step that you have crossed. And then you move on to non-clinical PK/PD and finally clinical. Now in clinical also, it's interesting to note that the studies, as I told you, non-inferiority or equivalence. So now look at the typical study that we do, on the left-hand side, where you want to know whether the original reference biologic is different from current standard of care. Means, is it superior, is it better, whatever. So there, the null hypothesis is that there is no difference. Then you do the study, you test this hypothesis, and if you do find a difference that is statistically and hopefully clinically significant, then you can reject that null hypothesis and accept the alternative hypothesis saying that, yes, it is different and maybe it's better. Here with a biosimilar, it's just the opposite. It's like the null hypothesis, if I may call it so, is that there is a difference, and then you do the study. So it has to be adequately sample sized, calculated, not just 100 patients in each arm. That is the point I was trying to make with the Indian guidelines, that they should say you should calculate sample size, because sometimes what happens is if you just say 100, and it may not be adequately powered to pick out the difference, and the difference actually exists, but your study was not powered to pick out the difference, then obviously, you are approving something based on the finding that you think there's no difference, but actually there could be a difference. So all that I'm saying is, because of the fact that here you have to actually show that there is no difference, it's just the opposite, you need sample sizes of a certain, an adequate number. It just can't be 100 and then a phase 4 study in more than 200 or whatever. I don't think that's adequate. And this is the entire package, of course, for biosimilar insulin glargine. It's much more than this. It was not just a PK/PD and then the phase 3 studies in STRIDE 1 and 2. It, of course, began with a lot of CMC comparisons, then moving on to bioequivalence and even euglycemic hyperinsulinemic clamp studies done abroad. And all these studies, of course, done for the US, European Union, UK, Canada, Japan, Australia, and all the major markets that even the DCGI recognizes. So that's why I've just put in here no difference to no difference. And the difference is when actually there is no difference. So then I'm just thinking, I know there is no such term, but if in India you have a biosimilar, which has done all that and is approved abroad, world-class quality, world-class approvals, would it be okay to call it an original reference biosimilar against which the other biosimilar should be benchmarked? I know this is not going to be possible because in any case, a biosimilar is compared with the biologic and not with another biosimilar. But my point is if other biosimilars in India don't really have to do all this workup, then naturally this biosimilar seems to be a little different. I'm not saying it's better. I'm only saying it's a little different because one is the drug substance is made here. So we're not dependent on other countries for drug substance or API. Secondly, world-class approvals. And thirdly, of course, interchangeability. So at least some difference does exist, but whether one is better than the other can only be answered if you design, and you don't need to do it, but if you design a well-designed, adequately powered head-to-head study. So these are, of course, the different studies that were done all over the world, 7 trials, 1,915 patients, 20 countries, and then, of course, the approvals all over the world again. I'll just quickly show you. In STRIDE 1, of course, was in type 1 diabetes patients, but for biosimilarities or a typical parallel group design. So I won't get into the study, except the conclusion was that there was no difference between the original reference biologic and the biosimilar. Now, what you're seeing, MYL, stands for Mylan, and that's because Biocon has this partnership with Mylan, where Mylan does the clinical development for Biocon, but the product is the Biocon Biologics product. And whether it was an efficacy measured by HbA1c reduction, FPG, and also the amount of insulin required, safety in terms of hypoglycemia, severe, nocturnal, and anytime hypoglycemia, and obviously immunogenicity. There were no differences between the two arms, and the same was seen in STRIDE 2. In STRIDE 1 was, of course, with 560 patients. In STRIDE 2, in 558 patients. This was type 2 diabetes mellitus patients. In STRIDE 1 was a 1-year study. In STRIDE 2 was a 6-month study. But here again, no differences on all 3 parameters: efficacy, safety, and immunogenicity. But what I think we need to focus on a little more is in STRIDE 3, because that is the study which actually gave us the approval as far as interchangeability is concerned. Now look at this design. So you had type 1 diabetes patients from STRIDE 1 who were actually on reference insulin glargine for 52 weeks, and then they were randomized into 2 arms. Arm B is okay. You just continue with reference insulin glargine, which is Lantus plus Lispro, because these are type 1 patients. But when you look at arm A, this is where the interesting bit comes in. So they are then, switched to Biocon's insulin glargine plus Lispro for 12 weeks because HbA1c. And then they're switched back to reference insulin glargine or Lantus for again 12 weeks. And finally, again switched to the Biocon insulin glargine for another 12 weeks. So 36 weeks or 9 months. And then obviously one would look at all the 3 parameters. So here I'll actually show you some data in terms of the change in HbA1c from baseline to week 36, which was equivalent to the reference insulin glargine. Similarly, in terms of safety, hypoglycemia, anytime, nocturnal, as well as severe hypoglycemia, no differences. And of course, this is the most important, immunogenicity, because sometimes if the immunogenicity to some biosimilar is much higher than that of the original reference biologic, then it can impact efficacy, safety, and then the doctor may wish to switch the patient back to the original reference biologic. But sometimes that may not be useful, because the same antibodies which destroyed the biosimilar can also destroy the biologic. Now, this may not be very often seen in clinical practice, but it is something that is theoretically possible. So we need to keep that in mind. And that is why it's extremely important to see these cross-reactive anti-drug antibodies in terms of percent binding between the treatment groups at any time point. And this was, of course, seen in all the studies in STRIDE 1, 2, as well as in STRIDE 3. So ultimately, the conclusion again was that no differences in efficacy, safety, or immunogenicity between the original reference insulin glargine and the biosimilar insulin glargine from Biocon. And therefore, the US FDA, I mean, normally Janet Woodcock would not be so effusive. She doesn't talk that much, but she actually said this. Today's approval, July 28, 2021, the first time it has happened in the world, and that too for an Indian company. Today's approval of the first interchangeable biosimilar product furthers FDA's long-standing commitment to support a competitive marketplace for biological products and ultimately empowers patients by helping to increase access. So we go back to the IDF theme about, can I improve access? And this is one way to improve access to safe, effective, and high quality medications at potentially lower cost. She's actually used the word safe, whereas in the industry, we very rarely use the word safe. And if we use the word safe, we always qualify it, at least on promotional materials. You can't use the word safe just like that, but maybe a regulator can use it. So this is my last slide. If you want to see what are the differences as far as Biocon Biologics Limited is concerned, I believe that the difference lies in our DNA, and that DNA can stand for data-driven, novel, even if we come out with biosimilars, but we do have a few novel biologics as well, and of course, accessible. You know, Kiran Mazumdar-Shaw believes that if you call a drug a blockbuster, it is not just a drug which makes $1 billion in the first full year of sales. She believes that if 1 billion patients can access your product, then it is a blockbuster. Thank you.
Thank you, Dr. Viraj, for this very exhaustive and informative session. I do see some questions from the chat box. I think there are probably a couple of comments and then a couple of questions as well. Maybe to kickstart. So I have a quick question about the InStride 3. Was there PK data also collected and analyzed and assessed from regulators as part of interchangeability? Patient PK.
As a part of InStride 3, I'm not sure whether it was collected, but apart from InStride 3, there was a study which actually looked at PK and PD. Okay. But as part of InStride 3, as part of the interchangeability exercise, I'll just check. I don't know if Pavan Kumar is still there on this call. Maybe he may know, but otherwise I will check with Dr. Sandeep Athale and come back to you. But a good question, yeah.
Yeah, no worries. And there is one question from Sujay Patil. Do we need to do separate interchangeable studies, or it can be done in combination or along with the design in a phase 3, a typical phase 3 study? And then his question also has a second part. Our prescribability and switchability studies are different.
Yeah. Now, my understanding is that you have to do a separate interchangeability study if you want to claim interchangeability, because the usual studies that we do, they prove that your product is biosimilar, which means it's prescribable. But see, what you can do is both the prescribable or biosimilar study as well as this interchangeable study, both are part of phase 3, but you can't really club it in one study. You have to do a separate thing because that's a 3-switch design. So that will give you interchangeability. And then, of course, you do your regular biosimilar kind of comparison studies, the typical parallel group, and then claim biosimilarity or prescribability.
Right. And then there is one question from Sushmit Sekar. He says, excellent presentation, of course, and US FDA guidance states that if a company believes that a drug can be designated interchangeable without clinical interchangeability study, they should provide adequate justification towards this belief. Your thoughts, what can be such situations where this can apply, and examples if there are any?
First of all, I am not aware of this, so I will have to read the US FDA guidance. I thought that US FDA is very clear that if you have to be designated interchangeable, you have to do a clinical interchangeability study. Now, what I was trying to tell you all is that this interchangeability designation is given exclusively to that company for 1 year. Now, this year, suppose Eli Lilly decides to come out with their own BIOSIMILAR, and then they approach the US FDA and they want to get interchangeability. What at least Dr. Sandeep Pathalia has told me is they will just get it. So I was a little surprised that why should they just get it without having to do a kind of, InStride 3 like study. But he said that is the, rule, because maybe, again, the whole point of this biosimilar discussion is that we want more and more people, patients, to have access. So therefore, as long as you can show that you are a biosimilar, perhaps after this 1 year period of exclusivity that maybe one company has got for its interchangeable biosimilar, other companies can just, so that's what maybe you're saying, that you have to provide adequate justification. So I'm wondering what kind of adequate justification will the other companies provide? Maybe they will try and say that, I mean, this is what Shubhadeep also mentioned, that, ultimately when you compare a biosimilar with an original reference biologic, it is the CMC data that really, really is important. Once you can show that, it's like taken to mean that even clinically, there could be some differences here and there, but those differences may not be as clinically meaningful as compared to if you make that company now do another InStride 3 like study, which means there will be some delay perhaps in that drug coming to market. So which means access can be an issue. And secondly, I don't know about the pricing differential. So therefore, I think what regulators want is that even biosimilars should be within the reach of as many indicated patients as possible. I mean, when I was in Boehringer Ingelheim, I would say the theme was we have to maximize the number of appropriate patients who should be on Boehringer's products. Now, what they meant was appropriate. What they meant, obviously, is per the label. So my question to them was, okay, all that is fine, but then there are many patients who are appropriate per the label who cannot access because the price is such, and therefore they can't experience the value of your innovation. So their theme was value through innovation. But how will they experience that value unless that innovation they can access? So that was the point I was trying to make, that I fully know, I fully agree that it takes a lot of effort, time and money. And I'm not saying that the R&D cost determines the pricing of your product, because R&D cost is considered sunk cost. So people look at what is called as anticipated income stream, and that in turn depends on your years of exclusivity left, how clinically differentiable are you, and is the market willing to pay that price? And then you determine your pricing based on other factors in the market as well. You do maybe a pricing research, etc. But my point is, if you've developed an innovation for patients, you have to somehow, I'm not saying only the company, it could be society, it could be the government, could be other players, philanthropists. We have to try and make the product as affordable as possible so that patients experience the value.
Yeah, no, well said, Dr. Viraj. So Melvin, there are, I think, another three more questions. How are we doing on time? Can we take them, or shall we take it?
You can take another 5 more minutes. You can take.
Okay. So then the next question I see here is it's about the sample size. What is the basis for sample size calculation for interchangeability studies, especially with respect to InStride 3?
So I know what you may be thinking is that InStride 1 had 560, InStride 2 had 558, but that's because they're pure, how do I put it, parallel arm studies. If you saw InStride 3, there is a parallel arm design, but if you look at arm A, it is the same patient. It's within the same patient you're doing a back and forth. So which means it's almost like using the patient as his own control. So like a crossover design, if you want to call it. And then naturally the sample size requirement would be lesser, because now you're looking at variability within the individual rather than between different sets of people. So that's why the sample size for InStride 3 was only 127 and not the 558 or 560 you saw. But otherwise, the way sample size is calculated I just look at it from delta, sigma, alpha, beta, and then you keep in some, for the dropouts, you look at the background incidence. I mean, the sample size calculation is more or less the same. I mean, the same, I would say, principles or parameters. But naturally here, the whole point is there should be no difference. Whereas your earlier other studies, which want to look at superiority and say that this drug is better than standard of care, etc. There you want to be able to show a difference. Here, the whole point is there should be no difference. So from that perspective, the sample size is calculated.
Okay. And there is one more question from the same delegate, Dr. Santosh Shiv. Are there specific challenges with respect to interchangeability studies for different diseases like diabetes and oncology? Because to do switch study in oncology might be a difficult one.
No, I fully agree. And that's perhaps one reason why our biosimilars in oncology, trastuzumab or bevacizumab, currently we only have them as biosimilars. So that's a good question for me to go back and ask Kiran Mazumdar-Shaw, that why can't we do interchangeability also for those two biosimilars? And perhaps it is as what you mentioned. But the way I see it is, ultimately, it's like this. Now, Shubhadeep also has mentioned it in the chat box. It's only in the US where you really, I mean, if you want interchangeability and if you want the pharmacist to substitute, then you need to do it. But in other parts of the world, at least till now, the regulators have not really included interchangeability. For them, as long as you can show your biosimilar, it's fine. Now it's up to the doctor to decide whether he can switch a patient who's currently on an original reference biologic to that biosimilar, because that data he doesn't have. He only has the data that they are biosimilar. But then again, if you go back to the US FDA, you realize that after 1 year, even the US FDA doesn't require that second or third or fourth company to do an InStride 3-like design. So which means maybe interchangeability is not that great, point of differentiation, because as you're saying in oncology, I'm sure in oncology also you can do it. It's not that you can't. It's just that, do I really want to be that sure before I switch? Because on the other hand, if I can get more of my patients to access this biosimilar, previously only very few patients could access that original reference biologic, who knows, maybe that is more important for me. And the fact that they'll be more compliant with therapy, because the cost is lesser. So that is where I would, means as a doctor for my patients.
Yeah, no, thank you for that answer. So in the interest of time, there is one last question, but I probably will park that because that's a very India specific question. So Maulik is going to talk about the Indian regulations. So probably, we can take this in Maulik's session or at the end of the panel discussion. But I'll just read out the question just for everyone's information. The question is, in India, do we have periodic studies for batch variations in biosimilars? So let's keep that question and come back to that when we are in the next session or probably in the panel discussion stage. So with that, once again, thank you, Dr. Viraj, for this extensive, exhaustive presentation and very interactive slot as well. So Melvin and Indumati, back to you now.
I thank both the speakers and the chairpersons for making the session most impressive